Okay, we will start in a few minutes.
Esempio, certificato mandato 20-28 febbraio. Senti.
Okay. Hello, everybody. I'm Marco Emili, and together with Fiorenza Stagni as chair of the Education and Training Committee. We are very pleased to welcome all of you to this webinar entitled The European ICOD Project Preliminary Evidence of Efficacy on Adaptive Functionality and Cognitive from Phase I/II Study in Adults with Down Syndrome. This webinar will be presented by Dr. Filippo Caraci and Dr. Rafael della Torre. The first speaker will be Dr. Filippo Caraci. He is an assistant professor of pharmacology at the University of Catania. During his career, he has published more than 250 papers in peer-reviewed journals, and he served as co-editor of several international journals. Dr. Caraci has worked in drugs discovery in depression and Alzheimer's disease, with the aim of identifying new pharmacological targets. He is current chief of the unit of neuropharmacology and translational neurosciences at the ERCCS Oasis Research Institute in Troina, Italy. And he is involved as coordinator of dissemination activities in the Horizon 2020 funded ECOD project. The second speaker will be Rafael della Torre. He is a professor of toxicology in the Department of Medicine and Life Science of Pompey Fabra University. He has published more than 600 papers in international journals, and his research focusing on the interaction between nutrition and cognition in different diseases, such as eating disorder, obesity, genetic and neurodegenerative disease, and on pharmacological and non-pharmacological approaches to improve cognitive performance in intellectual disability, such as Down syndrome and Fragile X syndrome. He is involved in many international projects, including the ICOD project. The duration of this webinar will be approximately 45 minutes, followed by 50 minutes dedicated to the discussion. For any question, you may write them in the chat, or at the end of the webinar, you may raise your hand to ask your question directly to the speaker. So if you want, you can activate subtitle in your preferred language. So, Filippo, if you want, you can share your screen.
Yes, Marco, thank you for your wonderful presentation. And it's a great pleasure to be here. Let me thank my coordinator, Rafael della Torre of the European ICOD project, and also Pier Vincenzo Piazza and Elis Pharma and all the team, because as you can see, today we will discuss not only about the new clinical data, but first of all, what we are going to do in the next months. So, what about cognitive deficit in Down syndrome? Big problem for families, because there isn't an approved therapy for the Down syndrome-related cognitive dysfunction. And the problem is not only present during, not only in young, but also in older, and the risk of Alzheimer's disease. A big problem in terms of diagnostic is to dissecting age-related cognitive decline from the underlying intellectual disability. And also, I would say the lack of international consensus on validating and accepted the cognitive psychometric tools. So when we start to evaluate a new drug, we should also to consider the new psychometric tools and new validated tools to understand and to evaluate the real efficacy of a new drug. Mainly, a deficit in working memory and executive function seems to affect functional autonomy in Down syndrome people. This point should be clarified now, because we have worked for many years to identify new pharmacological targets to treat working memory and executive function that significantly can affect functional autonomy. The European ICOD project, Improving Cognition in Down Syndrome, aims to address these unmet clinical needs, these real clinical needs, as when we discuss with families, by bringing into humans a new drug, an innovative drug, that targets with a completely new mechanism of action, the CB1 receptor, the receptor for cannabinoids. Because there is evidence of an hyperactivity of this receptor in Down syndrome, not only in animal models of Down syndrome, but in Down syndrome people, Down syndrome individuals. So these drugs developed by Elis Pharma, AEF0217, belongs to a new pharmacological class named signaling specific inhibitors of the CB1 receptor. Why I'm going to describe the name of the drug and the mechanism? Because the innovative mechanism of action will explain also the great safety. Because the first questions that I have when we discuss with families, okay, this is the mechanism of action, but what about safety? And the approach is completely new, because we have evidence that the drug works in animal models, but as you will see by the Rafael lecture, great evidence comes now also from humans. So, which are the partners of the ICOD project? The principal investigator, Rafael della Torre, which is, I would say, the greatest expert in the world in Down syndrome in cognitive deficits. And we had the pleasure to collaborate with Rafael, because Elis Pharma developed the drug. I worked in Bordeaux for 2 years, also contributing to the developing of this class, but all this work comes from Pier Vincenzo Piazza, Stéphanie Monleson, and I will tell you more because these are the other partners of the project. OASI Troina, where I work, in collaboration with the University of Catania, the Institute Jérôme Lejeune, Saint-Étienne, and a big thank also to the Hospital de la Princesa. You will see also the relevance of the partners in the data from Rafael. So the aims of the project is not only to develop a new drug. This point should be clear, and I have discussed this point several times in Italy with Scienza in Contra Down. The most relevant point is to develop, okay, the drug, a safe drug for treat cognitive deficit, but also the dissemination of objective. I'm the coordinator of the dissemination activities. What European Commission, what our monitor ask for us is to raise awareness on the impact of pharmacological research in treatment of cognition, but to improve also the diagnosis of cognitive deficit in Down syndrome. Here you can see the first page of our ICOD website, where I will show you some data that can be found in 5 languages: English, French, Italian, Spanish, and Catalan. Families can find a specific section to describe the project, which are the cognitive deficits, what are the definitions. One of the main problems that we have as neuroscientists is communication. Be clear, simple with families, not so many words, and according to the model of Cicerone, Tacito, and the Latin literature, language should be clear. And it's very difficult to explain the mechanism of action of these drugs, but can also be simple if we want. I will do my best after many contacts with Down syndrome families and association that I want to thank, and want to thank the Italian Down syndrome task force, coordinated by Lucio Nicci, because in these last 3 years, after many meetings, probably we have improved communication with families, because we know that individuals with Down syndrome have these difficulties, and mainly executive function in working memory, the inhibitory control. But this is a big problem that in the pyramid of unmet needs probably was a critical point when our project was approved. Because bring a new class for treat cognitive deficit in Down syndrome is a revolutionary, I would say, only if we combine pharmacological approach with non-pharmacological approach. The message of the project, okay, the recruitment, the study, the clinical study, but this big relevant aim can be reached, and is only with the collaboration with Down Syndrome Association. I would like to thank you, my association in Italy, the IPD, the Core Down, that helped me so much in adhering to this objective of the European Disability Strategy, that we have also had the opportunity to discuss in Brussels 1 year ago, and because this is our aim. Here you can see Stephanie and Monlejeune and Pier Vincenzo Piazza, CEO of Eris Pharma, and the story of the drug is a story of where technology transfer meet translational pharmacology, because when you develop a drug, you must start from great revolutionary points, and I will try to summarize these points before to leave the floor to Rafael, that I want to thank, as I want to thank all the other partners of the project, particularly Diego Real de Asua, because the Hospital Universitario de la Princesa in Madrid was essential for the Phase I study, and my institute and the University of Catania, Institut Jérôme Lejeune, and also the Centre Hospitalier with Relatori in Saint-Étienne. What about the endocannabinoid system? So the question is, why this target in Down syndrome? This is the question that I received from many families. Why you are discussing about this system? Because the endocannabinoid system is essential in synaptic plasticity. It's okay, but only recently we know that endocannabinoids such as 2-arachidiolglycerol and anandamide are involved in cognitive function, but are very high in Down syndrome individuals and activate in particular the CB1 receptor. So the question is, okay, there is an hyperactive cannabinoid receptor in Down syndrome, but there are also high levels of these endocannabinoids. Endocannabinoids are essential for our synaptic plasticity, for our cognitive function. But are too much, when the levels are too much, this hyperactivation can create some problem. Why? And please, I want to thank you, Alice Pharma, for the possibility to show this cartoon that was initially prepared to the Nature Medicine paper for another drug of the same class that is studied in other pathologies related to CB1 receptor. CB1 receptor is the main receptor of the endocannabinoid system, is involved in memory, has a clear role probably in the pathophysiology not only of cognitive deficit, but also in autism spectrum disorders, in psychosis, because the overactivation of CB1 receptor is involved in psychosis in schizophrenia. But what about the role, the physiological role of this receptor? The physiological role, as I many times discussed with Pierre V, and that I want to thank again, is to help to forget. Because normal cognition is a balance between remembering and forgetting. And during evolution, forgetting negative experience increased our capabilities to explore hostile environments. So inability to forget is essential to survive. The question is, when you have a CB1 receptor hyperactive, which are the consequences? You have an impairment. You have an impairment of cognition and memory. And this can be present in many pathologies, not only in Down syndrome. So ideally, inhibition of CB1 receptor can be a strategy, because you can, if you reduce the hyperactivity, you reduce the cognitive impairment. In the past, several antagonists have been studied, among which rimonabant. But this is not a great strategy to block completely a receptor, because the receptor needs to work for many functions. You have to block only the negative function related to cognitive impairment. And as you will see, this problem has been solved with this new drug. The evidence in animal model is that blocking the CB1 receptor rescue cognitive deficit, is essential to treat cognitive deficit. When I discuss about this clinical data, this is an excellent paper from the group of Andrés Osaita. We must consider that mice are different from humans. You will say, we know. But when you use long-term treatment in mice, you can have also great information in humans. Because if you treat mice for 4 months, 4 months in mice are equivalent to 13 years in humans, 10 months in mice, 45 years in humans. The question may be for you, but what about preclinical studies? We don't believe to preclinical studies. No, preclinical studies are essential, because if you study a new drug with a chronic treatment in mice, this is the basis that regulatory agency wants to have to go then in humans. And so the translational pharmacology is the pharmacology that try to link preclinical evidence with clinical evidence. So, which is the advantage with this innovative drug? That you have a lot of 2-arachidonylglycerol, as you can see, and you have an hyperactivation of the receptor. In this wonderful cartoon, you see the two different signaling pathways of the receptor, one linked to the G protein, one to the beta-arrestin. I don't want to go to the details, but the strategy cannot be to block all the signaling, but to block with our drug only the hyperactive signaling linked to cognitive deficits, and leave the CB1 working, as you can see in red, for the other physiological function. This is a completely novel approach discovered by Pierre V. Piazza 10 years ago, because these are derived from pregnenolone. I would say, considering Hippocrates and Greek language, drug mimicking nature. When a drug born from nature, from mimicking a molecular mechanism such as pregnenolone, you have, in general, this is a rule of pharmacology, a very safe drug. This was the genial intuition from Pierre that 10 years ago I had the opportunity to meet in Bordeaux when I was visiting professor in Bordeaux. So you have this selective inhibition that can explain the safety, not only the great efficacy. And this story can be found in different cartoons, but I will short and summarize to be more clear. So the innovative mechanism of action, you will see Pierre Piazza is to block the signaling related to cognitive dysfunction. And the evidence of preclinical efficacy has been confirmed not only in trisomic mice, we would say animal models with trisomy where working memory deficit have been well corrected, and the drug is able to reverse memory impairment, but as you will see in the next minutes, clinical evidence recently come from clinical study. So now I will leave the floor to Rafael della Torre, and if we have time, I need just 5 minutes at the end of after Rafael lecture, just to show you what we have done on the social, on the communication style. I would say the website, and first of all, LinkedIn, Facebook, where you can find all this information. So thank you again. I will stop here.
Rafael, sorry. The microphone is off. Okay.
Dicevo che grazie mille per l'invito a partecipare in questo webinar. I appreciate very much your invitation to participate in this webinar this afternoon. I'm going to talk essentially about the clinical development of the AEF0217 that was already started describing my colleague Filippo Caraci. Concerning the preclinical development of this drug, we almost finished with everything. There are some pending experiments on carcinogenicity in vivo and on C14 metabolism of the drug, but essentially this drug, from the preclinical point of view, it has a very high safety index. Therapeutic index is 650. Just for your reference, for example, paracetamol has a therapeutic index of 10, and this drug has a therapeutic index of 650, and this is because essentially the the drug mimics a brain mechanism of defense when there is an hyperactivation of the cannabinoid system. The other interesting issue regarding this drug in the preclinical domain is that none of the adverse effects which are associated to CB1 antagonists are observed in experimental animal models. So the other thing that is maybe relevant to you to know is that we are in a very advanced pharmaceutical development. The drug can be synthesized in 5 synthesis steps with low-cost components. The final formulation to be administered in clinical trials, particularly in the Phase II clinical trials, is already available. It's a drinkable formulation, which is, I think, nice and interesting for subjects with Down syndrome that sometimes they have problems with swallowing some drugs, particularly some capsules, for example. Then we've already, of course, we finished with the Phase I studies in healthy subjects. I will give users some details, and then also I'm interested in presenting to you some data on the Phase I/II clinical trials we performed in Down syndrome. Regarding the Phase I program in healthy subjects, where there were 68 individuals involved, what we did is what the regulatory agencies are requiring. I mean, we have to do a single dose and multiple dose ascending, single and multiple dose ascending studies, and then a food effect study to evaluate any interference on the viability because food. And the results, in short, is that no serious or severe adverse effects are observed. Only 3 effects related to molecular observed, mild diarrhea. There are no changes in biological markers, electrocardiogram or vital signs, and there are no differences in psychometric tests, different scales of depression, anxiety, psychosis, and suicidal tendencies. And globally, the drug in healthy subjects has a very favorable safety and pharmacokinetic profile. When we finished with the Phase I studies, we wanted to replicate in some way these studies in Down syndrome. We wanted to make sure that the drug was also safe and well tolerated in individuals with Down syndrome. And we undertook a clinical study that initially was really targeting these 2 objectives, the safety and tolerability. And we recruited 29 subjects in 2 sites in Spain, in the Hospital La Princesa de Madrid, and in my institution, the Hospital Research Institute. And these subjects participate in a double-blind control with placebo clinical trial with a single dose, 0.2 milligrams of the drug, that was administered once daily for 4 weeks. The profile of the individuals that participated in this Phase I/II study were young adults with Down syndrome with an age range between 18 and 35. And they were individuals with mild to moderate disability following IQ. 75% of the participants have a moderate disability, and 25% of the participants have a mild one. And as I told you, primarily, we wanted to evaluate the safety and tolerability, but and also the pharmacokinetics, because we were interested at looking at if there were some interference in the viability in subjects with Down syndrome due to the gastrointestinal motility of these subjects. But then we made this study that should be an easy study just to look at safety and tolerability, like the ones that we conducted in healthy subjects. We wanted to also to examine some indices of efficacy in terms of pharmacodynamics, and then in this context we evaluate also the adaptive functionality and the cognitive performance of subjects. And also we did some electroencephalographic registers related to cognition. And also we were interested at evaluating some biomarkers of drug target engagement, focusing on some C1 dependent ECG activities. Concerning the safety and the tolerability, in fact, what we did in short is to replicate what we observed in healthy subjects. The drug is very well tolerated, no safety concerns were identified, no serious or severe adverse effects were observed, nor did any one of the participants leave the trial because of the adverse effects. Most of adverse effects were mild in severity and were considered unrelated to the treatment. The type of frequency of emerging effects was similar in the placebo and in the active drug group. And again, there were no observation of the typical side effects of CB1 antagonists like rimonabant in the subjects treated with the drug. But then, as I told you, we wanted to examine if there were some indices of that the drug was active. And the first thing that we evaluated was functional adaptive functionality. We used this instrument, the BAPS, the Vineland Adaptive Behavior Scale, that essentially what evaluates our three different domains in subjects with Down syndrome. There is a communication domain where we evaluate the receptive, expressive, and written capabilities of communicating. Also, the everyday skills at the personal level, community level, and domestic level. And finally, there is a third socialization domain that looks at the interpersonal relationships, time to play and have fun, and finally, skills to handle difficult situations. We start with this functional adaptive functionality evaluation because at present, the European Medicines Agency considers that you are able to demonstrate an improvement in this adaptive functionality. This should be sufficient for the approval of the treatment. Then the priority today for the European Medicines Agency is adaptive functionality and not cognition like it was in the past. When you look at the results, what is interesting is that really the drug-induced improvements in all adaptive functionality domains. We have statistical significant effects in expressive communication, tendency in the written communication, improvements in the everyday scales at the personal level and the community level, and also in the socialization domain, in the interpersonal relationships. And as you will see, the difference between the group of subjects that were treated with this low dose of 0.2 milligrams of the AEF0217 versus the ones that were treated with placebo are quite different. We don't need to make much statistics to show the difference between both treatments. Then we were interested in looking at cognition, and for this we use a tool that is going to be used for the first time in Down syndrome clinical trials, which is the NIH Toolbox Cognitive Battery. That is a tool that was developed by the National Institute of Health in the United States. That in fact is a tool that genetically wanted to evaluate the cognitive performance of subjects, among other areas, on subjects in the whole lifespan of subjects, from the infancy to the age to the elder. But a group of clinicians, particularly clinicians that were involved in Fragile X syndrome, they in some way modified this NIH Toolbox Cognitive Battery, and now we have an NIH Toolbox Cognitive Battery for intellectual disabilities. And we were interested essentially in evaluating the fluid cognitive score of this battery. And what we do is we examine 5 different cognitive functions: working memory, cognitive flexibility, attention, episodic memory, and processing speed. We were in particular interested in the cognitive flexibility, considering the preclinical data we have. And the idea is that from the European Medicines Agency, any improvement in cognition is considered supportive evidence for the improvements on the adaptive functionality. In addition to administering this cognitive battery, we performed some electroencephalographic evaluation of subjects while they were performing a working memory task. That was not the same working memory task, as you can imagine, that the one that we evaluated with the NAS toolbox. The working memory task we were evaluating was from the Cantab battery. And we were interested at looking at the resources that use the people with Down syndrome to solve these tasks. Typically, the people with Down syndrome, they have difficulties to solve the task, and they use, let's say, more resources on your brain than the neurotypical population. When you look at the results on cognition, we have almost very, very close to statistical significance in the cognitive flexibility. But what's really interesting was the experiments we performed with electroencephalography while the subjects were performing these working memory tasks, because we were able to reverse the increase in EEG activity during the task, which after the administration of the drug, we reached levels of activity that we observed in neurotypical control. That means that the activity in the brain of people treated with the drug is the same that for neurotypical subjects to solve this working task. And these were really very nice results. We were also interested at evaluation target engagement, because again, this requirement for the regulatory agencies, and particularly from the European Medicines Agencies, you have to be able to demonstrate that your drug is really acting on the brain, and acting on the brain in areas which are relevant for the mechanism of action of the compound you're evaluating. And from previous studies that we performed in neurotypical subjects and subjects with Down syndrome, we observed that people with Down syndrome, they have an EEG profile which is very similar to the one you observe when somebody has been consuming THC, tetrahydrocannabinol. And then we develop a software specifically to evaluate EEG responses that are CB1 dependent. And the interesting thing again is that here we observe a large effect on the gamma intertrial coherence during auditory steady response at 40 hertz. I know for you it's something meaningful, but these gamma bands are particularly active in the hippocampus in the brain, which is related to many of our cognitive functions. Then we believe that we have a CB1 indication that there is a target engagement. And if you look at the results again, you don't need much statistics to compare the activity you observe in the placebo group and in the group of people that are treated with the drug. Then the summary of results of these phase 1-2 studies, in people with Down syndrome, is that the AEF0217 has a very favorable safety and pharmacokinetic profile, because it has been very, very well tolerated. There were no safety issues. The compound was very well absorbed, and there were not large differences with what we observe in healthy subjects when they are administered with the drug. And we have some indications that there are some signs of effectiveness, because we observed improvements in adaptive functionality, in cognition, and probably the most interesting thing is that you have to consider that the intervention just lasts 4 weeks, and in 4 weeks we were able to observe this difference, which is very, very nice. And the other thing is that we have clear signs of target engagement, which are related to what I showed you previously on the involvement of CB1 the involvement of the CB1 receptors in the effects of AEF0217 based on 17 based on the EEG activity of the subjects. Then now, what's next? We have to move to the phase II studies in subjects with Down syndrome. And what we are planning is a multi-center international phase II clinical trial, 2B clinical trial. There will be 188 participants with Down syndrome with an age range between 16 and 32 years of age. There will be 10 sites participating in this multi-center clinical trial. On your right, you have the centers involved in Spain, France, and Italy that were participating in the phase II studies. The idea is that we're going to test 3 doses of the AEF0217, 0.1, 0.2, which is the one that we tested in the phase I/II study, and 0.6 milligrams per day versus placebo in a double-blind design. Here, the intervention will last not 4 weeks, but 6 months. And of course, the main objective is to confirm the improvement in adaptive behaviors observed in the phase I/II studies and identifying the best suited dose of AEF0217. And the idea is we are expecting to start in March 2026. I mean, it's tomorrow. The thing is that we are pending on some final confirmation from the TETIs that we can start with the randomization of subjects. The design is that, as you can see, there are 4 arms. 3 correspond to 3 different doses of AEF0217, and one is the placebo. The randomization will be in a ratio 1:1:1:1. And as you can see, the intervention will last 24 weeks, and there will be afterwards a period of follow-up of 8 weeks. As I told you, we are targeting subjects with 16 to 32 years, with a BMI between 18-35, full trisomy 21, an IQ between 35 and 70, able to hear and to communicate with evaluators, stable illnesses with stable medication. There are very few restrictions in terms of the comorbidities of subjects that can participate in this study. There were some restrictions on some drugs, particularly some drugs that may interfere with the cognitive evaluation. We are expecting that these subjects have clinical chemistry test in the acceptable range, as well as ECGG, vital signs, and everything that fits with reality. And also, it's very important to have a caregiver who lives with the participant and can participate in the evaluations, because still a large part of the evaluation of the efficacy of the drugs is on the hands of caregivers, and that they provide a lot of information of what's going on. The primary objective will be to demonstrate that there is an improvement in adaptive behavior after the administration of the drug when compared to placebo. And then we have secondary objectives. One concern efficacy. We want to demonstrate an improvement in cognitive function, quality of life, and sleep efficiency. Of course, there is always the issue of safety, but it's not really a major concern. But it's true that it's going to be the first time that we administer the drug, not only for weeks, but 24 weeks. We are going to collect additional data on the pharmacokinetics, and there will be some exploratory pharmacodynamic objectives. One regards the concentrations of the major endocannabinoids to acetylglycerol and anandamide, and the other one is the potential effects of AEF0217 on body weight and parameters related to glucose and lipid metabolism. And that's it. That's a short summary of where we are in the clinical development of this drug. Filippo.
Rafael, thank you. There are 2 wonderful questions from Jean de la Barre and And Brian, if you want to answer to this question, then I can leave 5 minutes for communication, as you want. But I think it's relevant to answer to Jean, because Jean Labat said, very interesting results, Rafael. Some years ago, we have shown that DIRC1A overexpression is impairing CB1 receptor functionality. And this could be modified with JCL. Do you think 184? Do you think that this could be related to your results?
Frankly speaking, I don't know, because I'm not familiar with the GCL 184 drug.
If Jean wants to add Jean, Marco, if you want, you are the chair, so I don't want to.
Jean, if you want to explain your question, we have time.
Yeah, well, hello. That's a result we had which has been published in a paper with the first author was Tommaso some years ago, and so maybe you can have a look at that, and you will see if it's relevant or not.
Yeah. No, but of course, I mean, the fact that D1A that we have been working with when we were working with epigallocatechin interferes with the CB1 receptor functionality. This could be interesting. We were not looking really at D1A activity this time in our clinical trials. And regarding the question to Brian Stotko, yes, the idea is that we start, of course, communications with the European Medicine Agency, particularly because also we have been developing a pediatric plan also for the development of the drug immediately after finishing the phase II study. But yes, we are planning to start communication with the FDA for further studies, maybe for phase II or phase III studies. We'll see.
We have another question from Natalia. Is CB1 expressed by immune cells?
To my knowledge, yes.
Yes.
And do you foresee a future clinical trial in children?
Yes, this is the priority, because in fact, Down syndrome is a neurodevelopmental disorder, and theoretically, the earlier you start in your intervention, you have more chances of improvement. Then children are the priority, but we have already an approved pediatric investigation plan approved by the European Medicine Agency to apply it immediately after finishing the phase II studies.
Other question? I have a question. Have you or do you intend to evaluate some biomarker in the plasma, maybe, for example, for the neuroinflammation, because I know that CB1 family receptor are implicated in the immune response, so maybe I don't know.
We have some plans to investigate some microRNAs, yes, in plasma.
In plasma microRNAs, okay.
Yeah.
There is an interesting question from Natalia about the effects on the immune system. Rafael, I think that we can add to the story the fact that we are studying also the link between CB1 receptor and TGF-beta 1. We have published two papers. Margherita Grasso is the first name of this work we done with Rafael and the team from Torino. There is some evidence that overactivation of CB1 receptor can reduce the anti-inflammatory cytokines. TGF-beta1 is an anti-inflammatory cytokines, very useful for cognition. And in the future, we would like also to study the link between not only CB1 receptor and TGF-beta1, but also the link in terms of molecular impact on immune cells.
Vincenzo?
Yeah.
Vincenzo Militello,
Vincenzo,
yes.
please. Yeah.
You're muted.
If you want, Vincenzo, you
Thank
can
you.
do
Thank you.
the
Thank you.
question as you want, in English or in Italian, as you prefer.
I try to English, but I hope there is understandable. So very, very simple. As father of a Down young, there is a best frame for the age of these people, Down people that are in the champion group in the first phase of this proof. And also there is a IQ frame, there is the best solution as target. Thanks.
As I told you before, these environmental disorder, then the earlier you start in your interventions, the better. But I think that probably in the future we have to think in drugs combined with some lifestyles interventions regarding diet and also exercise and other things that are very much relevant in the development of individuals and cognitive performance, not only the drug. I mean, the magic bullets doesn't exist. They help. They contribute to improving the intellectual disability, but it's not the solution at all.
Thanks.
Okay.
We have another question from Marco.
Yes.
Yeah. There are numerous studies that demonstrating the link between neurodegeneration in individuals with Down syndrome and oxidative stress. Could this drug be helpful, considering that CB1 appears to be associated with neuroinflammation? In this can be possible. How?
That's a good question, because when we were planning the phase 2 study, we wanted to as you observe that the range of age of people participating in the phase 2 study will be between 16 and 32. To our, let's say, surprise, the European Medicines Agency, they wanted to have young adolescents participating also in the study, because at the beginning we were proposing 18 years instead of starting at 16. And for us, it's fine. I mean, but then we proposed to, on the other hand, to have an upper range of age larger than 32, and they say no, 32, because if subjects participating in the study, they start to have neurodegeneration, then we are going to mix the cognitive disability with the neurodegenerative process, and then they say 32, although we thought that we have tools today to know if at 35 or even older ages, any neurodegeneration, there were signs of neurodegeneration, but we were limited to do our work at 32. But in the same way that one of the first targets will be to perform these studies in pediatric population, the second one is to start with people at risk of developing neurodegeneration. That means that probably we have to start with subjects in the early 20s, and to see if we, in some way, we can prevent in some way to improve the neurodegenerative process. Yeah.
If I can add a comment, Rafael, the other hope is that TGF-beta 1 is reduced in Alzheimer's disease and in Down syndrome with Alzheimer's disease.
Yeah.
We are interested to study also the link after the 32 years. But again, what I say to families when we are going to recruit, I have 34, we are starting to work also on the biomarkers over 32, over 33, because, okay, the ICOD trial start now with these rules, but we hope that to extend in pediatric field as clearly stated by Rafael, but also in a longitudinal way in Down syndrome individuals. We hope in that have an high risk to develop Alzheimer's disease, not only as you have clearly stated, but the regulatory pathway is long. We should start with the first nucleus and step by step as okay. If we have 3 minutes, I would like to give.
Sorry, we have another question.
Okay. I'm sorry.
Why mosaicism are not included in the trial? I mean, Natalia.
Great question.
Well,
No,
typically
Natalia.
because they have cognitive performance that is higher than in the full trisomy subjects, and then also it depends on the mosaicism, which is the extent of the mosaicism, and then of course anyway if the drug works for the full trisomy is going to work for the mosaicism, then there is no problem for that. But in most clinical trials with people with Down syndrome, you typically the ones with a very low IQ, because you have difficulties to communicate with them, or the ones with mosaicism are typically excluded as extreme cases in these clinical trials.
Okay. Okay, Filippo, if you want to.
Thank you. Just 5 minutes to show where they can find the updates about the project. Just 5 minutes, because there is an ICOD section for scientists and an ICOD section for families. So I will show very rapidly. I don't want to go behind what expected. I don't know how can eliminate this one, but unfortunately the subtitle works only in English, I found, not in Italian. Okay, we will go again to about communication, disseminate the results. We have a right dissemination board. We have a logo. We have a Down syndrome patients-centered research, because we want to put families. This is our logo, this is our flyer, but this is our website, where you have an ICOD for families. ICOD for you is the ICOD for families. Simple information, all updates. And I would like to thank you, Salvo Calacciura, from my institution for the constant update in Italian, but also BCA from Spain, from and Cristina Jimenez for updating the website every day, or we'll say every week. In the website, you can see what we have done. What we have done now, we have communicate with families. We have a X, I would say Twitter at the beginning, now X, because we're born in 2021, but we have also Facebook. We have LinkedIn. LinkedIn probably is the most relevant site for us in terms of update, but also Facebook is very useful. And you can find the post with the updates about what our lecture. For example, in the news and the research section, you can find also the lecture. I will have from Marco, if you agree, from the T21, the video of this lecture. I would like to put it on the YouTube ICOD channel. ICOD channel dedicated to ICOD project, where we will describe in the future of the mechanism of action. Also, we would like to increase the use of the ICOD channel to the ICOD Twitter account, LinkedIn, and Facebook. Yeah, if you want, go on the ICOD channel, you can find different video. For example, with the same content discussed today, for example, discussed in Syracuse, in Sicily, or in Barcelona, or in France, in different languages. Okay, and I think that also the X sites. I don't like so much X sites as Twitter in the past, but it's very relevant. In the ICOD project, in the YouTube channel, you now can find the Scienza Incontra Down, all the, for example, you have the video in Italian and in English, where we have summarized the different content of recent results. So you can, and also from the Italian Down Syndrome Task Force. I love this image because it's our first ICOD symposium in Troina with 100 families and present in Troina. Troina is in the center of Sicily, is the first city of the Normandian reign. This is the program in Madrid where we have done the second ICOD symposium with Ospedale Princesa. I would like to thank you again, Monica Enzini, our officer, that helps us so much in supporting also the project, and also the Spanish society, the different association that Rafael de la Torre has involved, from Fundació Catalana down in Spain and all the other ones. The good from my side is that I can say that in Italy we have a great organized Italian task force working very well, that includes the organization, Down syndrome organization, the Italian, the two main ones. And this helps us to communicate about the clinical trial, and also to share, for example, lecture from Pier Vincenzo Piazza on the mechanism of action of these drugs, to better explain the innovative mechanism of action. I'm also interested as pharmacologist to study in the project the link between the patient's reported outcome and the impact on functional autonomy. What I would like to say that is the first time that a new drug has been studied of putting functional autonomy for an innovative drug as one of the first outcomes from EMA. Perspectives and further steps include participants with Down syndrome in our communication boards to better understand the needs of Down syndrome individuals. Here you can find some the mail where you can contact me, but again, we will improve the ICOD YouTube channel. The new phase 2 studies is going to start. This is my contact as coordinator for the dissemination activity. And in our clinical trial also will be involved gemelli with Angelo Carfie and Dr. Pirazzoli and Luisa Sambati from Bologna, and other center from Bordeaux, and this is my group team in Troina that I would like to thank you for the support. But again, what I can tell and what I'd like to conclude, what is the best of this project is that our relationship with families, with the Down Syndrome Association. I organized with Rafael and the other colleagues 33 different meetings in these last 4 years. This has changed my life, at least my cognitive style and my possibility to communicate with Down syndrome families and to understand the needs of these families and to improve also the treatment of cognitive deficit in Down syndrome. Thank you again. I'm stopping here.
Okay, so thank you, Rafael. Thank you, Filippo, for this interesting webinar. And thank you, all of you, for the attendance. And see you in the next T21RS webinar. See you.
See you then.
Thank you so much to
Thank you.
T21.
Thank you.
Thank
Thank
you
you for
so
your
much
time.
to all and bye.
Grazie Vincenzo. Thank
Grazie
you
a te
to all.
dell'interessante occasione di riflessione e di conoscenza.
Thank you.